Hi, I'm Becca,
I'm Chris.
and this is our bacteria experiment. Our other member of our group, Victoria, is not here, so we have to take over her parts. And Chris, take it away with the question and hypothesis.
Okay, so our question is, how does the amount of meat in different varieties of dog kibble affect the bacteria levels found on a piece of kibble? This question is interesting to us because we would like to make a recommendation on what kibble has the least amount of bacteria on it. We will use 3 different types of dog kibble with 3 different levels of meat in the kibble and ham that we are using. We think that there will be more meat in the dog kibble and more bacteria that there is to be bacteria. To count the bacteria, we need a precise number, which will help in our graphs.
This is our expected graph of what we predict the percentage of meat in the dog food will be. We think that a plain Q-tip will obviously have 0% meat. A Nova brand dog food will have 10, BG will have 50%, and ham 100%. This is our expected combined graph of meat and number of colonies. We think that 100% meat will have the most amount of colonies. This is our expected graph for expected colonies. We think, again, that 100% meat will have the most colonies. We think that the percentage of meat in each type of dog food will affect the level of bacteria because the more meat, the more bacteria that survive the cooking process. We think the bacteria colonies will increase as the percentage of meat in the dog kibble increases, and a piece of ham will have the most bacteria colonies, and our negative control will have the least amount of bacteria colonies. This is our combined research paper. The main points that we found online were that bacteria like E. coli and Salmonella can survive the cooking process and can harm or kill or sicken our dogs, and that humans are 4% more likely of getting the Salmonella or E. coli infections if they keep dog food in the house. Our sources were rawfed.com, bornfreeusa.org, and thedogpress.com. Our materials show that we needed sterile Q-tips from our science teacher, Sauber, for swabbing samples, petri dishes from Sauber for growing the samples, media from Sauber as food for the bacteria, parafilm from Sauber to securely seal the plates, dog food from Chris and our friend Mikey for testing, and a camera from Sauber or Victoria to record data by photo evidence.
Okay, so our procedure is to sterilize the surface, and then we collect all the materials, Q-tips, plates with media. We have to make sure the plate's not contaminated or that ruins our experiment, parafilm, etc. We need to label the plates on the bottom, date, anything to identify it. Then we need to take the samples and streak and inoculate the plate in each quadrant. We need to repeat that 4 times or in as many areas in the plate as we need to. When finished, we parafilm the plate. Then we will take a photograph and we place it in the incubator and repeat those steps as many times.
For as many tracks.
Yes, or as many
This is our data table done by Chris.
Okay, so we have 4 trials, then our averages. Our negative control is the no dog food, and it pretty much had no dog kibble except for the last trial. It came out to be 72, and that was very unexpected. So
The plate was probably contaminated.
actually, I don't think it was. Our first dog kibble that we tested was from our friend Mikey again, and it first one was 1 bacteria colony. Second trial was 3. The third trial was 12, and the fourth trial was 57, which was a very large jump. And then our average was 24.33333. Okay. Then our dog kibble number 2 from my dog's kibble was 2 colonies. Then we couldn't count the second one because it was contaminated with the first dog food. Then our third trial was 16, fourth was 97, huge spike, and that was unexpected. Then our average was 38.3333. Then our fourth dog food, as you can see, was ham, and it surprisingly had 0 for 3 of our trials. And then the last one, it had 150, so our average was 50.
This is our not our expected graph, our actual graph with our actual data. As we expected, ham did actually produce the most bacteria, just as we expected. There aren't a lot of points that are actually in the best fit line, if there were to be one, which would probably be from maybe there through there through there. It was really sad that there were a lot of outliers, and that might mean that our averages aren't exactly reliable, but the percentages seen on the graph are not the actual percentages. Our actual percentages was for a plain Q-tip, 0%. For before grain dog food, 32% meat. Our Innova dog food was 92%, and ham was obviously 100%. So that was very interesting. But it shows that in trial 2, it did not show any data for a negative control Innova or for ham. So that was kind of strange. These are photos of one of our plates where the agar was poured by Sauber, but the plate was inoculated by us. In the next 2 pictures, we both poured the agar and inoculated the plates. And in the 4th picture, Sauber again poured the agar, but we inoculated the plates. And that is our bacteria experiment from 2010.
Hope you liked it.
Yep. And it's too bad Victoria wasn't here to be part of this.
Yep.
Goodbye. I pressed done.